Director of Machine Learning at Recursion. Recursion is a full-stack drug discovery company, started close to 10 years ago. We actually have a 10-year anniversary party happening in Salt Lake today. The company went public, I think it was 2019, like in the middle of the pandemic. The strategy is based on using a lot of machine learning and generating a lot of preclinical data in-house. And by now we have a handful of programs that are already in phase 2. So we've made a lot of progress in the last 10 years. My talk today is pretty technical. It's focused on the work my team is doing. But at the end, I can take questions on opportunities of the company in general. So what I'll talk about today is a technique called high content screening, which is a method for automatically generating a lot of images from cell-based assays. And then the second half of the talk will be more focused on how we analyze that kind of data. So this is what high content screening looks like. You can take 3D4, 1536 well plates, grow cells in them, and then expose them to different perturbations. This can be compounds like drugs, can be genetic perturbations, these days mostly using CRISPR. And then we can get images of how the cells are responding to those types of perturbations. So traditionally, when academic or industry labs use microscopy, it's been like lower throughput, kind of validating that a certain protein is being affected by like a specific treatment. But this approach really scales that up to say, okay, what if we screen an entire drug library of like 1 million compounds on a certain disease model? Or it can be also used for functional genetics, like let's knock out every single gene and see how the cells are responding. So you end up with a massive cell image-based data. So that's when you work with this approach. So I can describe a few use cases. So one that's a bit more intuitive is drug screening. So in this case, you can have a disease model. So our company published a preprint on COVID, I think it was also back in 2021, where you can just simply take the COVID virus and then apply it to the cells, and you can see the cells kind of changing in their structure and morphology. And you can also image the cells without the virus, so just a healthy control. And then if you do a drug screen, you can try to see which drugs are making the disease model look a lot more close to the healthy state. So the preprint, we looked at a handful of compounds that were going into clinical trials, and we predicted which ones would actually be effective in clinical trials. So I think we got like 7 out of the 9 accurate. So it kind of shows the power of this technique. So yeah, in this setting, you're kind of more or less converting the images to like a similarity search-based problem. The problem with this, from a drug discovery perspective, is you kind of need to repeat the screen every time there's a new disease model. So you'd have to image your like millions of compounds every time you want to like, look at a different type of disease. So an approach we've switched to is using the same technology for decoding biology. So in this case, we, in an upfront fashion, do large-scale perturbation screens, and then use the representations we get of the imaging experiments to kind of relate them to each other. So you can imagine this in the genome-wide setting. If we knock out every single gene with CRISPR and then do this kind of assay, we can kind of find interactions between genes. So if 2 genes have a similar effect on the cells, like a similar phenotype, that could be a really interesting insight for the preclinical researchers. So this example shows the mTOR pathway. So there's some complexes that have a similar, like play a similar role in the pathway, and then there's some inhibitory steps there too. So when we go to just looking at cosine-based similarity of the embeddings that we get from these imaging-based experiments, you can see that like TSC1 and TSC2 are like pretty similar in space, and that they have an opposite effect of mTOR. So you're almost kind of naively discovering these pathways just by building your data set in this manner. So these 2 use cases motivate why we use high content screening. And I'll talk about how we analyze this data, because that's kind of the big chunk of work that we've done at Recursion. So this is kind of the traditional way to do it. It's still used a lot. So it's based on segmentation and feature extraction-based approaches. And there's an open source software called Cell Profiler that's really popular for doing this. There's also a bunch of industry versions of the same process. But this requires you to look at the images and then write algorithms that can segment every single cell individually from the background and from each other. And then there's a whole bunch of mathematical functions you can apply to those regions to get measurements from each cell. So those are things like shape, size of the cell, texture features, like how bright the different markers are in the experiment. And then you end up with a big feature matrix where every single row is a cell, and then the columns would be like different types of measurements. Some of the problems that come up with this is like it's really challenging to get a robust segmentation algorithm. Like you can do the screen once and then get accurate results, and then you do the screen again and something shifted in the microscope a bit, and now like all the segmentations are wrong. So it makes everything downstream of that really unreliable. Also, the features are, just texture or shape features that are like manually described, so they could be leaving a lot of signal like outside of the analysis, and a lot of them are also correlated with each other. So there's a lot of additional steps that you need to do to like even leverage the features after the feature extraction. So like the rest of the world, like the field's kind of transitioned to using deep learning over the last 5 to 10 years. So I'll talk about a few different approaches to using deep learning for this kind of data sets. And the first is just directly transfer learning. So essentially we're working with images. There's lots of off-the-shelf imaging, like image deep neural networks that you can download that were trained on natural images like the large-scale ImageNet data set. So here I'm showing some results from really well-known benchmark data set in the field where you're just trying to group compounds into mechanism of action. So that's the accuracy that you're seeing there, like 90% and 91%. But this is comparing the feature extraction methods to literally just taking an ImageNet model and then extracting features from the cell images given an ImageNet model and scoring them. So you can see you can get roughly the same or even better accuracy with a model that had nothing to do with cell images, but it can recover like most of the structure that's important for scoring how compounds are affecting these cells. So as people in machine learning, we're kind of dissatisfied with that. Like we want to train models that are, kind of tuned to the specific data sets we're working on. Oh, sorry, this is one more result. So this is another model from Google that was also trained on natural imaging data. So they developed just a different kind of normalization technique that made that model even better. So on the same problem, we get 97% accuracy with a model that's never seen any cell data. So again, like, these models are really powerful even without adapting them to cells. But like I said, yeah, we do want to adapt these to cells because the assumption is that we'd get even better models. So this is a Kaggle competition that Recursion ran in 2019, and it was affiliated with NeurIPS. So here we released a relatively large data set that had 100,000 microscopy images. Sorry, 125,000 microscopy images, mostly with silencing RNA treatments, so siRNA. So this is a method to silence genes in assays. Like it's really noisy, so it's not used too often these days. But the data set had about 1,100 treatments, and the challenge was to learn a model that could predict what the treatment was. And then we held out a few experimental batches. So the winning result had I think 99% something accuracy on predicting the actual treatment in the held out experiments. And the person who won the competition actually joined Recursion within a year. So he's been working at Recursion for a while. Just last year, we finally got around to releasing a paper on that model and on that data set. So this was presented at a computer vision workshop this year, I guess, earlier this year. And this is what the data set looks like. So a big focus of that challenge was dealing with batch effects, because when you do these kind of experiments, you get a lot of artifacts that come up, even if you repeat the exact same experiment, just because of differences in the lab equipment or whoever's conducting the assay. So what you see here is like 2 different examples of the treatments, but every column is a different batch. And the data set had 50 different batches of the exact same 1,100 treatments. These are the channels that we typically image. So this is based on a protocol called CellPaint, where we look at 6 different morphological stains in cells. So like nuclei is the obvious one, but there's some other ones in there. And all our assays are standardized on these 6 stains. That's like the imaging format I talked about. That's a breakdown of the data set. So a few control conditions, but roughly the challenge was to predict the 1,100 siRNAs. The data set had different cell types too. So for the most part, it was HUVEC, but we had some other cell lines. And this is the approach that won the competition. So it's still based on training supervised models. And you train the supervised deep learning models to predict the treatment. And then at test time, if you want to get features for a new treatment, you pretty much use the model all the way up to the classification layer. So it's like the one layer before the prediction is what we use to, what we call like embed the imaging data. So that's typically just a vector of 128 dimensions. It's not really interpretable, so we can't say like, this first feature means that, but we can like, we see that they have really good downstream performance on a lot of different benchmarks. About a year after deploying that model, we updated it with a method called adaptive batch norm. So this might be too much detail, but the deep learning models use a technique called batch normalization. So each layer is normalized across the samples that are in the batch. But our experiments are also kind of batched by experimental batches. So if you combine those two methods and kind of leave this mechanism on even when you're inferring at test time, then you can actually get some property that makes the model more robust to differences in different batches. So you can see the results there, but basically this approach makes the model more robust to like held out samples or like held out batches. But yeah, switching gears, the past like 8-9 months, we've been working on a newer technique that's based on self-supervised deep learning. So these approaches have really kind of changed the paradigm in deep learning and in machine learning in general. So basically the whole field's moving away from transfer learning from big supervised data sets with lots of labels to self-supervised learning with even bigger data sets that are unlabeled. So if I go back to the ImageNet example, like in that case, you might have like a lot of images that are annotated as having like a cat or a dog, and you train your model to predict what's in the image. With unsupervised methods, you just give the model all your data without any labels, and there's different types of mechanisms where the model can like learn good patterns just based on the input data itself. So for biological use cases, this is really powerful because a lot of times we don't really trust our annotations or they're expensive to generate. So just using the raw data directly from our microscopes is a really powerful technique. So the results I'm calling out here are more for like the standard natural imaging baselines, but Google in 2019 released a big model that was trained on 300 million labeled images, and then their accuracy on this benchmark was 87.5. And then this one came out in 2021, this self-supervised one, trained without any labels on only 100 million images, ends up getting 87.8%. So even better performance with a lot less labels and a lot less training data. So the main techniques people use are masked input models. So for language, these are the kind of pre-training tasks that power large language models like GPT, like the GPT models. Essentially during training, like one of the earlier tasks these models are trained to do are predict masked words in a sentence. And that's really easy to train because you can take a huge corpus of text and just mask out different ratios of words. And what the model is actually learning in this step is kind of just patterns in natural language. Obviously, there's more that goes into GPT, like reinforcement learning with human feedback and like other types of techniques, but this is kind of the baseline model that they start with. More recently, researchers at Meta have proposed this masked autoencoder, which takes the exact same approach but applies it to images. So this is what the model looks like. You take an image and then you mask out different patches of it. You encode only the visible patches, and then you try to predict the missing patches. So this kind of pattern lets the model learn a lot of relationships that just occur naturally in images. So like in the original paper, there was like images of a bus where like all the model sees is like a wheel and like a yellow pixel, and then it can kind of reconstruct the rest of the bus. So that's kind of the idea here. So yeah, during this year, we spent a lot of time implementing these models, and we train them on cell data. So you can see here, might be hard to see if you're sitting far away, but you can see what the masking and reconstructions look like. So in this case, the middle column would be the reconstruction, and then the column on the right is the original image. So you can see, like aside from some color variance, like we see a lot of fidelity between the two. And the input to the model is this the first masked image on the left. So it's pretty amazing that given like so little visual information, the models can do such a good job like filling in the missing pixels. This is cool, but not really useful on its own. So we also have to benchmark these models. In terms of data sets, we also grew the data sets quite significantly. So the RxRx1 paper I was talking about had 125,000 images. Last year, we released a data set called RxRx3. So this is a genome-wide screen plus a few thousand compounds in a specific cell line. We didn't make all the gene knockouts public, so they're just kind of masked identifiers. But this data set has roughly like 2 million images in it, and that one's publicly available. But we also scaled up the training to like 53 and 95 million data sets that were built internally from pretty much like a lot of what we've generated to date. So like the reconstructions are nice, but we actually need to benchmark these models. So the way we do this is based on a paper we published in 2021 called, it was called Biological Cartography, but the idea is like, how do we score the relationships that we find with these models? So if you go back to the example I talked about earlier with mTOR, like we basically want to show that the model is discovering as much known biology as possible, and also discovering unknown biology, like that we can't really benchmark because it's unknown. But the way we use these models is we get the embeddings from them, so the features from them. We do a few normalizations to correct for batch effects. And then we use cosine similarity to relate how similar different treatments are. So different genes and compounds, like different compounds and compounds. And then we curate a bunch of known relationships from public databases. So there's 5 that we look at, things like Reactome, Signor, like you might have heard of some of these. And then we just see, based on the top 5% and the lowest 5% of our predicted relationship scores, how many relationships we recall from known biology. So that's roughly the benchmark we're looking at. So here's some recall metrics. So you're looking at how many relationships are recalled for different thresholds on the relationship scores. So normally we use like this one as a reference, the 5 and 95% one. But you can see here is that the self-supervised model on the 95 million data set is performing the best by far. And then the supervised model, which initially was trained on the smaller data set, is actually doing better than the supervised model on the larger data set. So this was our big motivation for exploring self-supervised learning in the first place. And it has to do with just issues with like labeling the supervised data sets. By default, we just assume that every single treatment's having an effect, and that's how we label the models when we're training supervised models. But that kind of breaks down when you scale to like hundreds of thousands of perturbations and like close to 100 million images, because those relationships are just, those labels are just too unreliable. Like a lot of the treatments might not be doing anything. Maybe they have artifacts. Maybe they're similar to each other, and we're saying that they're different. But when you use the self-supervised learning approaches, it's just learning on the images themselves. So there's no kind of bias introduced to the model. This is a table format of the same results, but you can see if we look at the different databases that we're comparing against, by far, yeah, the largest masked autoencoder model is doing the best compared to everything else. And another interesting feature of these models is that they scale really well to larger data sets. So I showed that when you use supervised learning, if you go to a bigger data set, the performance can actually get worse on a lot of biological benchmarks. But here we started with a data set that was around 2 million images, and we scaled all the way up to 95 million images. And then the model size itself, we scaled from about 20 million parameters to, I think it was like the biggest one has like 700 million parameters. So the models can get a lot bigger, the data set size can get a lot bigger, and the performance only continues to improve. So this is very much in parallel to like the results we're seeing with natural language and natural images, where bigger models, bigger data sets often result in better performance. So it's a nice regime to be in for biological data. So yeah, I'll end with that. In terms of careers, like there's one opening on my team, so we're continuing to do this work, but starting to incorporate other modalities. So far it's been really focused on images and microscopy, but we're starting to look at gene expression type data sets that we're collecting. We also acquired 2 companies this year that are based in Canada, so Valence and Cyclica, that have a lot of expertise in digital chemistry. So we want to combine kind of better representations of chemical perturbations in these types of models as well. And we have quite a big footprint in Canada. So about 2 years ago, Recursion announced that they were launching sites here. So we have a big office at Queen and Spadina that can host 50 plus people. And there's also an office in Montreal, like in the Mila building. So that's the Valence, what we're calling Valence Labs. So it's a bit separate from like the main Recursion entity, but they're doing a lot of research that's a bit more forward-looking. And in terms of other roles, we also have a huge data science organization, so that's kind of split between the headquarters and Toronto. So if you look on the site, there might be roles that are just data science roles. And on the clinical side, that I'm less familiar with, so I apologize if there's attendees interested in those positions. But if you see anything online, feel free to reach out to me if you have questions.